I said good morning all of you. Good. This is what the energy should be. This is second hour only. And if people have gone lower level of glucose. You all have glucose at your brain cells, neurons? Yes or no? Fine. Okay. No more wastage of time. We shall start. We have to cover little more today. What was the last part that I ended up with? Glycolysis. We have ended till this was the substrate product, isn't it? From glyceraldehyde 3-phosphate, what we got is the first oxidative phosphorylation reaction. Where did we end it? We have covered till formation of 1,3-bisphosphate, yes or no? What was the previous to this reaction? Glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. I have elaborated this example, this reaction as no, conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Oxidative phosphorylation, where you are going to see a formation of a reduced equivalent that is called as NADH. Okay, that's the only oxidation reduction reaction provided by the enzyme called as glyceraldehyde 3-phosphate dehydrogenase. Am I clear to all of you? Am I clear? Okay. And apart from that, it being a dehydrogenation reaction, we have also seen that an inorganic phosphate has been incorporated. So because inorganic phosphate is getting incorporated, the enzyme name is not given as kinases. Is it clear to all of you? Okay. Moving to the next reaction, where 1,3-bisphosphoglycerate converts into 3-phosphoglycerate. Just look at the substrate and the product and name the difference. Name the difference. Be fast. One phosphate has been released out. Yes or no? If a phosphate is getting released out, there should be someone to carry it. Yes or no? Who should be the carrier? ADP. Very good. And formation of an ATP. Okay. This is the first substrate level phosphorylation reaction in the process of glycolysis. But I said this is the first substrate level phosphorylation reaction of glycolysis. What is substrate level phosphorylation? In general, the formation of ATP is a total functionality of mitochondria, where the reducing equivalents goes inside the mitochondria. Okay, they adhere to some complexes of electron transport chain, and at the end, at the terminal part, is what oxygen is consumed by the fourth complex, that is called as cytochrome oxidase. Okay, after that, because of some change in electrochemical potential, some gradient is formed in the inner membrane space. Okay, regarding that, the complex 5 is responsible for production of ATP. This is the whole scenario. We will go through that. Okay, basically, the formation of ATP is a complete process provided by mitochondria. But if a molecule of ATP or GTP or ITP is getting formed at substrate level, at the level of cytoplasm or any other organelle without the involvement of mitochondria, we call those sort of reactions as substrate level phosphorylation. Direct production of ATP. Okay, these substrate level phosphorylation reactions plays a very important role in organelles or in cells which lacks mitochondria. Sorry, not organelles, in those cells which lacks mitochondria. For example, matured RBC. Immatured RBC contains mitochondria, but matured RBC lack mitochondria. Okay, the perfect example that we can give for the energy product provided in this sort of cells is provided by substrate level phosphorylation. Okay, apart from that, this reaction plays a very important role in RBC also. Regarding what? See, there is an alternate cycle called as Rappaport-Lewberin cycle. मैं short में लिख रहा हूँ. It is called as Rappaport-Lewberin cycle, that is responsible for the formation of a component called as 2,3-BPG. What is that? 2,3-bisphosphoglycerate. Okay, in general terminology, in the general sequence, the substrate is 1,3-bisphosphoglycerate, it converts into 3-phosphoglycerate. in general glycolysis. Where it is happening? Only in RBC. Where? Only in RBC. Only in RBC. Okay. But in specific RBCs, this 1,3-BPG can also convert to 2,3-BPG on certain stimulus. Fine. And the stimulus is hypoxia or lower concentration of oxygen. Lower oxygen saturation. Okay? This hemoglobin has got a beautiful tendency to keep or to save, oxygen within itself. Because hemoglobin has got a higher affinity for oxygen in general. Okay? Hemoglobin has got higher affinity for oxygen. What does it mean? If hemoglobin is having higher affinity, that means it should be certain condition only when the hemoglobin is going to unload the oxygen. Yes or no? It should be certain condition. Fine. Now, you are under hypoxia. So, what you expect your hemoglobin should be performing? What do you expect? It should unload the oxygen so that your tissues get oxygenated. Yes or no? Yes or no? Right? But because of this higher affinity of hemoglobin for oxygen, hemoglobin is unable to unload oxygen to the specific tissues, resulting in hypoxia. Fine? So, under hypoxia, red blood cells have got a specific enzyme called as 1,3-BPG mutase. What is that called as? 1,3-BPG mutase. This is enzyme specifically present in RBCs only. Okay. What is the role of this enzyme? It provides the production of 1,3-BPG. It provides the conversion of 1,3-BPG into 2,3-BPG rather than converting it to 3-PG. Shutting down, shifting of reaction, dependent or provided on due to some stimulus. Okay. Once this component is produced inside the RBC, what it will do? It will go and bind to an allosteric site of hemoglobin. ध्यान से सुनिएगा. It will go and bind to an allosteric site of hemoglobin that will cause the decrease in affinity of hemoglobin for oxygen. As a result of that, due to the binding of 2,3-BPG, this allosteric effector, okay, hemoglobin will unload the oxygen because affinity has been. Is that clear? So, hemoglobin having higher affinity for oxygen in general, but once 2,3-BPG binds as to the allosteric site, what happens that the oxygen leaves because the affinity of the hemoglobin for oxygen reduces. That cycle is specifically called as Rappaport-Liouvering cycle. This cycle or the production of this specific component has got a very important significance for the mountaineers. Once you reached at the peak, oxygen saturation dropped, you went under hypoxia, fall down. Okay, that's why the concentration of 2,3-BPG in the RBC provided in the persons or the people staying at higher altitude is very high as comparison to the people who are staying at base areas. This is the reason. Okay. Apart from that, if the condition is retrieved and the hypoxia goes, normal oxygen saturation reaches, the left out 2,3-BPG can convert back into 3-phosphoglycerate by an enzyme called as phosphatase. Okay, that removes the phosphate in the form of PI. Oh, is that clear? 3-PG converts into 2-PG. What is happening? 2-phosphoglycerate. 3-phosphoglycerate is converting into 2-phosphoglycerate. What is happening? Shifting of the phosphate group from the third carbon to the second carbon. What is happening? It is nothing but an isomerase. Isn't it? It is an isomerase. What we call that isomerase basically? We call it as phosphoglyceromutase. Sorry. Shifting. It's just shiftment is occurring. Okay. Yes or no? Next is a very important reaction for the process of glycolysis, where the high energy bond, the high energy phosphate bond is restored. We do have phosphate over here, but the next reaction is conversion of 2-phosphoglycerate into a component called as phosphoenol pyruvate. The formation of this enol linkage or formation of this enol group is responsible for the next reaction that is going to release energy in the form of ATP. So the formation of enolic component over here is very important for the next step to release certain energy. Okay, and this is provided by an enzyme called as enolase. This enzyme again has got a very good significance for you can say laboratory medicine, for laboratory practices. Why? Because I have told this, I have given this example before also. Whenever we are going to estimate the blood glucose concentration, plasma glucose concentration, we collect the blood sample in a specific vacuum, in a specific tube. That tube is colored as grey. Remember, I have told this once. It is grey color. Okay, why this grey color? Because this vacuum contains an anticoagulant and a component called as fluoride. It contains a component called as fluoride. What is the role of fluoride? What is the role of fluoride? This enzyme requires a cofactor called as Mg2+ also. Okay? It requires a cofactor as a Mg2+. When we add fluoride provided to the blood sample, this fluoride chelates this Mg2+ and makes it not available for the enolase enzyme. As a result, the enolase enzyme becomes inactive. Is my point clear? Yes or no? Okay? As a result, the enolase enzyme becomes inactive. Because this enolase enzyme is getting inactivated, the glycolysis shut down, is closed, is blocked. So what will happen if you are blocking down the glycolysis? If you have collected blood sample, what are the cells you have? Blood cells. Blood cells are specifically dependent on the glucose provided in the tissue or in the blood cells. So if you are blocking, after collecting the blood from your capillaries or the veins, if you are adding fluoride to it, you are blocking down the glycolysis. Because you are blocking down the glycolysis, you are expected to get the true value of glucose present in your blood. Okay? For example, if you are performing glucose estimation without blocking the glycolysis pathway, you collected my blood now, you are going to perform the test after 1 hour. So within this time period, it is estimated that some amount of glucose will get utilized by the RBC for their energy requirement. As a result, what will happen? That the glucose level will fall down. That is a false falling down of glucose. This is a false result. So to get true result, that what was exactly inside my blood, when we have collected, we add fluoride so that it blocks the glycolysis, and as a result you get the true value of glucose present in your blood. Is that clear? Next reaction, where phosphoenol pyruvate is converted into a component called as pyruvate. You know this. Enzyme name is pyruvate kinase. Yes or no? If it is kinase, that means phosphate group is going to get transferred, and the component that is going to accept the phosphate group is ADP, that will convert into ATP. Can we call this reaction, can we call this step of the reaction as substrate level phosphorylation? Yes or no? Yes. So there are 2 substrate level phosphorylations that you are going to see, out of which one is an irreversible step. Do you remember I have told you that whenever I use red pen, just remember that that step is going to be important? Okay, so I have used. So in total for the entire pathway, how many times I have used red pen? 3 times. Reaction 1, reaction 3 and reaction 9. They are going to be very important in terms of regulation. Yes or no? Is that clear? This is the whole pathway. Okay. Now we are moving towards certain questions that we have arisen during while we were continuing with all these things. Okay. The first question, I have told you people, I have written it somewhere here. I have told you that make me remember about this question. What was that? Yes, lactic acid. That glycolysis takes place in two conditions, aerobic and anaerobic. Aerobic bole to presence of oxygen, where the product you are going to see pyruvate, but when it is in anaerobic, the product is lactate. Why? Anyone? Just some guesses. Anyone? You know this RBC contains hemoglobin? Yes or no? है ना? So hemoglobin should be reached with oxygen. So RBC is having oxygen, but still RBC has to depend on anaerobic glycolysis for the production of energy. Why? Anyone? बोलो. ये पकड़ दो ना को please बोलो. Say. No. Why compartmentalization of enzyme? Enzymes are already there in cytoplasm. Anyone? Any guesses? बोलो. To reduce the use of oxygen. No. Acha, say me one thing. Everywhere it is written, you people know we are surviving because of oxygen. Where is the maximum use of oxygen that we consume? Huh? क्या बोला? एक जन बोलो. We are consuming oxygen, yes or no? We are breathing in oxygen. Where is that oxygen, maximum percentage of oxygen getting consumed?
So breakdown of food and release of ATP.
No. Release of ATP. Where? Okay, fine. He gave you a hint. Huh? Mitochondria. Where mitochondria? Inner membrane. What in inner membrane is there which consumes the oxygen? ATPase. No. The fourth complex of electron transport chain. The fourth complex of electron transport chain is called as cytochrome oxidase, that incorporates the oxygen into H plus to form H2O. Okay, there is what the maximum consumption of oxygen, and ETC is working for what? To convert the reducing equivalents into oxidized equivalents. Like NADH should convert into NAD plus, FADH should convert into, FADH2 should convert into FAD plus. And as a result of this, we say na that 1 NADH equal to 2.5 ATP formed. बोलते हैं कि नहीं बोलते हैं? 1 NADH when is oxidized gives 2.5 moles of ATP. 1 FADH2 when oxidized give 1.5 moles of ATP. बोलते हैं कि नहीं बोलते हैं? This is what there is happening, and for the conversion of this reduced equivalent into ATP, okay, we require oxygen. Is that clear? Fine. So now my question was that what is making the RBCs to convert pyruvate into lactate? Yes. Suppose this is RBC. Does it have mitochondria? Yes or no? No. देखो मैं आंसर बता रहा हूँ. Significance of lactate formation. Why anaerobic glycolysis causes the formation of conversion of pyruvate into lactate? I'm giving a significance. Okay. Why? Because mitochondria is not there in RBC. Point number 1. ठीक है? So because mitochondria is not there, the NADH formed in the fifth reaction of glycolysis, will it convert into NAD plus? Yes or no? Just now I said, the conversion of reduced equivalents to oxidized equivalent is the whole total responsibility of mitochondria. Yes or no? So mitochondria is not there. Fine. In glycolysis, above this step, you have seen that one molecule of NAD plus is converted into one molecule of NADH plus H plus. देखा था कि नहीं देखा था? देखा था? है ना? And suppose करो this entire glycolysis is occurring inside the RBC. Fine. Imagine. In this state, if this is happening in RBC, is RBC having mitochondria? अरे बोलो यार. Yes or no? No. So if there is no mitochondria, this NADH that is formed in the cytoplasm, can it get converted into NAD plus? A yes or no? No. So as a result, inside the cell, inside the RBC, the ratio of NADH to NAD will increase. Yes or no? These all are organic components, coenzymes. They are not like present in unlimited abundance. No, they are present in limited form. That's why their cycling is important. Their cycling is important. If it is formed over here, it has to reconvert back into NAD plus. Otherwise, the glycolysis will shut down. Is that clear? So the concentration of NADH and these oxidized equivalents are limited. That's why cycling is required. And where this cycling takes place? It takes place in mitochondria. But RBC lacks mitochondria. So as a result, जब तक इसका concentration था cell के अंदर, the glycolysis will keep on going till there was enough concentration of NAD plus. Because for this reaction you require NAD plus to proceed with this reaction. So till the moment there was enough concentration of NAD plus, the glycolysis was going on. And the product was formed as NADH. And the end product was pyruvate. It is getting formed. There will be a moment when the concentration of NAD will fall down. There is no more NAD plus. No more NAD plus. As a result, the glycolysis has to shut down. It has to get blocked. Because this reaction requires NAD plus to proceed. Yes or no? Is that clear? Am I clear? So, to make sure that there should be enough NAD plus, to make sure that the cell have enough NAD plus to proceed, to continue with the glycolysis, so that the RBC will keep living or will be surviving, what they do? They convert the pyruvate into lactate. Fine. This is the reaction provided by an enzyme called as LDH, lactate dehydrogenase. Yes or no? Okay? This reaction requires a molecule of NADH plus H plus, and that converts into NAD plus. This reaction is happening in cytoplasm. Can you connect the link? Are you able to connect the link? This reaction, point number 1, is happening inside the cytoplasm. Point number 2, this reaction is utilizing the cytoplasmic concentration of NADH. Point number 3, this reaction is responsible for production of cytoplasmic NAD plus. Point, where is this NADH coming from in the cytoplasm? Sorry. Yes or no? So there is a correlation between the 5th reaction of glycolysis and this reaction, making it possible for the RBC to survive, so that the glycolysis should not stop. Because RBC is completely dependent on glycolysis for their survival, for the production of energy. Am I clear? Yes or no? Fine. So the NAD plus that is produced is reutilized in this pathway. So as a result, the process goes on. Is my point clear to all of you? Are you clear with the significance? Are you clear with the significance? Scenario number 2. This is what happening in RBC. I'm giving you a question. Okay. You have done strenuous exercise. You have gone through strenuous exercise. Strenuous exercise. Okay. Muscle cells, they do have mitochondria. Yes or no? Yes or no? They do have mitochondria. Okay. You are taking oxygen rapidly. Yes or no? But still, in rapidly contracting muscles or rapidly exercising, you are doing some heavy exercise and all, you feel muscle cramps after that. That is because of the formation of lactic acid. That is because of the formation of lactate. Fine. You are rich in oxygen. Your muscle cells have mitochondria, but still, why anaerobic glycolysis? You got the significance? This interlink? समझ में आया कि नहीं आया? इस लिंक का होने का मतलब यही है कि सेल के अंदर अगर ग्लाइकोलाइसिस को कंटिन्यू करना है, I'm saying this in Hindi because of few people only. अगर सेल ग्लाइकोलाइसिस को कंटिन्यू करना है तो we need a concentration, enough concentration of NAD+, and that enough concentration of NAD+ can be only provided by reconverting the NADH formed in the fifth reaction to NAD+, so that this NAD+ participates back in the fifth reaction and continues the glycolysis. Yes. So now, if this same thing is happening in skeletal muscle cells do have mitochondria. They are rich in mitochondria, but still anaerobic glycolysis is going on. Why?
It starts aerobic and the oxygen itself is still needed to make the glycolysis. So in order for the glycolysis to be done,
Fine, okay. But why you think that oxygen is getting lowered down in rapidly contracting muscles? Sit down. Yes, the condition is called as oxygen depth. It is called as oxygen depth. Fine. This oxygen depth is because of you are exercising. Your muscles are rapidly contracting. What you need? What your muscles need? Energy. Energy in terms of ATP. Yes or no? If you require ATP, if your muscle requires there is a heavy requirement of ATP, what the muscle cells will be doing? They will be having a hyperactivity of mitochondria. Yes or no? Yes or no? So for formation of excessive ATP, what you need? You need excessive oxygen. Yes or no? So as a result, the tissue concentration of oxygen will fall down. Yes or no? That is called as oxygen depth. Am I clear? This is because this is the reason for the oxygen depth or lowered level of concentration, lowered level of oxygen, lower saturation of oxygen. Am I clear? This is causing the formation of, this is causing the conversion of pyruvate to lactate. Okay? Just because oxygen has lowered down, the mitochondria has stopped functioning. Because mitochondria has stopped functioning, the cytoplasmic concentration of NADH is getting high. Lower concentration of NAD+. Because of that, it has to link with this reaction, so that the concentration of NAD+ remains enough inside the cytoplasm and the glycolysis keeps on moving. And our muscle cells never, gets lag of ATP or energy. Am I clear? Fine. This excessive formation of lactate in the skeletal muscles causes muscle cramps, or we can call it as the situation as lactic acid cramps also, accumulation of lactic acid, and this accumulation of lactic acid is converted back into glucose provided in liver. Okay, how? That is called as Cory's cycle. This is a very common question. This is a very common question in short notes. Justify the significance of Cory's cycle. So what is Cory's cycle is? Cory's cycle is the complete reutilization of lactate and conversion of it into glucose. It is a cycle that is occurring between two different parts of our body. Lactate formed in this muscle moves out into the capillaries out of the cell. Through the capillaries, it reaches the portal circulation. Through portal circulation, it goes inside the liver. Liver is responsible for conversion of non-carbohydrate precursors into glucose by the process called as gluconeogenesis. Liver cells are responsible for gluconeogenesis. As a result, the lactate moves out of the muscle cell, goes into the capillaries, into the blood supply, then moves back into the liver, where the lactate is reconverted into pyruvate. Pyruvate converts into glucose by the process called as gluconeogenesis. This glucose again goes into the blood. Through the blood, it reaches the muscle, and the muscle again becomes energized by the utilization of glucose. This cycle is called as Cory's cycle. Am I clear? Am I clear to you? Any doubts still here? Are you people okay with the significance? It's a very, tricky question they generally ask you. In short notes and, reasoning type questions, they will ask you. Just justify the formation of lactic acid, justify the significance of production of lactic acid under anaerobic condition. They will ask you. And as I have taught you this, I want the explanation in your paper like this only. Is that clear? Now, moving to regulation, the most important part. यहाँ पे थोड़ा से you need little clever acts. You need to be smart enough to understand regulation. And believe me, if you can understand over here how to go through regulation, I feel it for me itself. You don't need to go through the books to look for the, okay, what should be the regulators. You can say it by yourself only. If you follow this process, you have to be smart enough. Okay, see how. The first irreversible reaction is provided by hexokinase reaction or glucokinase in the liver. Fine. Is that clear? The first irreversible reaction, reaction number 1, I have written in red pen. Okay. Hexokinase, glucokinase. Hexokinase or glucokinase provided by hexokinase specifically is regulated by feedback inhibition. So what is the product of hexokinase? Glucose is converted into glucose 6-phosphate. So the product of hexokinase is glucose 6-phosphate. So once the concentration of glucose 6-phosphate is very high inside the cell, very high inside the cell, okay, it acts as a feedback inhibition. It acts as a feedback inhibitor of the enzyme called as hexokinase. Yes or no? Am I clear? Am I clear? Same stands for glucokinase also. Simple, feedback inhibition. And the inhibitor is higher concentration of glucose 6-phosphate. It is G, okay, not 6. Higher concentration of glucose 6-phosphate. Now coming to the most important, the bottleneck of glycolysis, the rate limiting step of glycolysis, that is what called as PFK1, phosphofructokinase 1. Bottleneck. ध्यान से सुनना, यहाँ पे थोड़ा hormonal regulation भी है, that is what covalent modification is all about. यहाँ पे थोड़ा allosteric regulation भी है, दोनों होने वाला है साथ में, ध्यान से देखिएगा, ठीक है? We see that the reaction stands as fructose 6-phosphate is converted into fructose 1,6-bisphosphate. This is what we generally see by the enzyme PFK1. PFK1. This is the enzyme that requires a molecule of ATP and converted into ADP. We require Mg2+. Yes or no? Fine. When we come to the regulation part, this enzyme, as I have told you in the previous class, that this is the rate limiting enzyme, this is the rate limiting step of the glycolysis. When I say rate limiting step of any of the pathway, what does it mean? It is going to be an allosteric enzyme. Yes or no? मैंने बोला है? Did I say that? Yes. Rate limiting step of an enzyme, just close your eyes and think about that, okay fine, this is going to be an allosteric enzyme. That means there should be some allosteric effectors of it. Either it can be positive effector, either it can be negative effector. Positive effector increases the efficiency of enzyme, negative effector decreases the efficiency of an enzyme. Yes or no? Clear? In general, we see the picture like this. Fructose 6-phosphate converts into fructose 1,6-bisphosphate. But before fructose 6-phosphate is broken down or is changed into fructose 1,6-bisphosphate provided by PFK1, something else is happening at a background, you know, at the background side. What is that happening? There is a bifunctional enzyme, what we call bifunctional enzyme. This bifunctional enzyme has got two enzymes, PFK2 and other is fructose 2,6-bisphosphatase. Am I clear? Am I clear? Imagine you had food. Imagine you had food. So your blood is flooded with glucose. Yes or no? So all the cells of your tissues, all the tissues should start utilizing the glucose. Yes or no? so that the glucose level in the blood should come down? Yes or no? Fine. As you had food, relevant to that, insulin got released. What? Insulin got released. Okay? And insulin is responsible for activation of an enzyme called as phosphatase. Phosphatase. Yes or no? Okay? Scenario number 2. Imagine you didn't have food. Fine, you are in fasting state. That causes the release of glucagon from the alpha cells of pancreas. Yes or no? Okay? And glucagon is responsible for release of an activation of an enzyme called as protein kinase A. In short form, we call it as pKa. ठीक है? एंजाइम का नाम देखो, काइनेज. क्या करेगा? फॉस्फेट ग्रुप को डोनेट करेगा, बाहर निकालेगा. And the donor or acceptor should be ATP or ADP respectively. Yes or no? Phosphatase. What should be the work of it? Removal of phosphate group in the form of inorganic phosphate. Fine. Coming to scenario number 2 first. You didn't have food. Glucagon is in blood. Causes the activation of an enzyme inside the cell named as protein kinase A. That is responsible for the addition of a phosphate group. This is what called as covalent modification. Why I am calling it as a bifunctional enzyme C? Imagine this is a lecture room. Next to that there is one more lecture room. Some other lecture class is also going on. Both the classes are functioning differently, but they are under one roof. Am I clear? That's why we are calling it as bifunctional enzyme. And the functionality of this enzyme is correspondent or reciprocal to that. अगर यहाँ पे फर्स्ट ईयर का क्लास चल रहा है तो क्या वहाँ पे फर्स्ट ईयर का क्लास चल सकता है? नहीं, वहाँ पे सेकेंड ईयर चल सकता है, वहाँ पे थर्ड ईयर चल सकता है. Reciprocal. Okay. In that way, when you didn't had food, glucagon was there, protein kinase A was activated, fine, and it causes the release attachment of a phosphate group over there in the surface, rooftop. कहीं पे भी लगा दिया phosphate group. That caused the addition of this phosphate group into this bifunctional enzyme, caused the activation of this enzyme and inactivation of this enzyme. Fine. Are we concerned about that now? No. Because this enzyme is inactivated. To continue with the glycolysis, this enzyme has to be activated. How? You had food. You had food. Insulin came into play. Insulin got released. Phosphatase enzyme becomes activated. This became activated. Phosphatase is the enzyme that is responsible for removal of phosphate group. Phosphatase removed the phosphate group in the form of Pi. Now, see the change. PFK2 becomes activated and this enzyme becomes inactivated. Yes or no? Reciprocal regulation. These two states cannot come at one single time. It has to be reciprocated. Fine. Either you can be in a well-starved, well-feeded state, or you can be in a fasting state. दोनों एक साथ नहीं हो सकता. Reciprocal regulation. This is what called as reciprocal regulation. Is that clear? Once this PFK2 is activated, this fructose 2,6-bisphosphate, which was waiting there to enter into this reaction, first goes to this reaction and converts into a product called as fructose 2,6-bisphosphate. ध्यान से सुनिएगा. Before this reaction has started, the fructose 6-phosphate was waiting there. Fine. Once this got activated, the fructose 6-phosphate immediately converted into fructose 2,6-bisphosphate for some time, not for lifetime, for some time, till the concentration of fructose 2,6-bisphosphate becomes very high inside the cytoplasm, unneededly high. Fine. This high concentration of fructose 2,6-bisphosphate act as an allosteric effector and will activate PFK1. The moment fructose 2,6-bisphosphate attaches to PFK1 at an allosteric site. You remember PFK1 is an allosteric enzyme. Yes or no? Fine. So the moment fructose 2,6-bisphosphate attaches to PFK1, the PFK1 becomes active. Once the PFK1 is active, glycolysis chalu. Yes or no, buddy? Yes or no? Am I clear? Am I clear to all of you? Fine. So the most important, the most potent allosteric effector, positive allosteric effector of glycolysis is fructose 2,6-bisphosphate. Yes or no? Most potent, most important. Is that clear? Apart from that, there are some other allosteric effectors. Dekho, you can guess it. See. Take a scenario. Let us say cell is having high level of ATP in one scenario. Cell is having low level of ATP and high level of ADP. This ratio should be like this. Okay, fine. How to understand? I told you, na, you are not supposed to, mane book खोलने का जरूरत भी नहीं पड़ेगा. If you go through this. Two scenarios. One scenario where concentration of ATP is high. Second scenario, concentration of ADP is low. Sorry. Scenario number one, where concentration of ATP is high and the concentration of ADP is low. Scenario number two, concentration of ATP is less and concentration of ADP is high. Now you say me, all of you are going to say that. Positive allosteric effector. Positive effector matlab it is going to activate PFK1. Out of these two, which one? Which ratio? Now for this, you have to think, you have to be smart. How come? Think about glycolysis is occurring for what? What is the need of glycolysis in the cell? Energy. Energy. तुम्हारा पेट भरा हुआ है, बाबा राम बने हुए तुम, ठीक है? पेट भर के खा लिया है, you're full of energy. मैं तुमको बोलेगा नहीं बेटा और खा, खाओगे? नहीं खाओगे. Same happens with the cell. If the cell is having lot of energy in terms of ATP, why do it will go for glycolysis? Yes or no? Do we need? No. So in that moment, which out of those two scenario will inactivate the glycolysis as an allosteric effector? Socho. Think about it. Which out of this scenario is going to act as a allosteric inhibitor? Which one? Scenario 1 or scenario 2? Scenario 1 or 2? I have already told the answer. That means you people are not smart. I told you that glycolysis is occurring inside the cell to provide energy for the production of ATP. Your cell is already having ATP. Why to go for glycolysis then? So the increased concentration of energy बोले तो increased concentration of ATP and lower concentration of ADP. Fine. दोनों एक साथ नहीं हो सकता. Reciprocal होना. Fine. In that scenario, if the cell is already having lot of energy, increased concentration of ATP, it is going to act as a negative regulator. That means it will inactivate PFK1. Sorry. It will inactivate PFK1 allosterically. Yes or no? Yes or no? Huh? Now the cell, scenario number 2, the cell is not having enough energy. भूख लगा हुआ है cell को energy का. It requires lot of ATP. That's why the concentration of ATP is low, low energy state of the cell. In that situation, lower concentration of ATP, or you can say ADP, higher concentration of ADP will act as a positive modifier of PFK1. It will activate the PFK1 positively. Am I clear to all of you? Fine. Apart from that, increased concentration of cytoplasmic citrate. Acha, citrate is a component of TCA cycle, yes or no? Is citrate present in cytoplasm? It's a component of TCA cycle. TCA cycle अपने components को बाहर जाने एक शर्त पे देता है कि जितना मैं बाहर भेज रहा हूँ उतना वापस भी आओगे. This is called as anaplerotic reactions of TCA cycle. The concentration should be equally matched. They are very valuable components, citrate, in the cytoplasm. So citrate coming into cytoplasm is a signal for something, and the signal is high energy state. When the cell is having lot of energy, again high energy state of the cell, TCA cycle shuts down. बंद हो जाता है TCA साइकिल। क्यों जो जबरदस्ती और ATP बनाना और ATP बनाना और ATP बनाना? So the citrate is sent out of mitochondria. So citrate present in cytoplasm. Remember this. This point is going to come for the regulation of fatty acid synthesis. Remember this. Okay. Citrate coming into the cytoplasm signals the cell that we have lot of energy in stock. We don't want any further energy. As a result of that, you should stop down the catabolic process and you should go with the anabolic process. As a result, the citrate act as a negative allosteric effector for PFK1. It inhibits glycolysis. Glycolysis is a catabolic pathway. Am I clear? The last one. Can I take 5 minutes more? Thank you. The last reaction. Pyruvate kinase. ये आप लोग बोलेंगे, मैं नहीं बोल. Is that clear to all of you? Again, commonly asked question. Reciprocal regulation. Explain the reciprocal regulation of glycolysis and gluconeogenesis. Common question. Is that clear to all of you? अगर नहीं समझ में आया तो एक बार बाद में अगर फिर भी पूछ लेना मेरे से. Now, the third one. That is called as PK. PK वो आमिर खान वाला नहीं, pyruvate kinase. ठीक है? This is active. This is inactive. Okay? Fine. PK is inactive when phosphate group is added to it. PK is active when no phosphate group is added to it. Now, say me, what should we write at the sides of those arrows, directions? When PK should be activated? बहुत सिंपल है बेटा, बहुत सिंपल है. सोचो ना, अभी थोड़ी देर पहले बोला, which is the hormone that is responsible to add phosphate group? When you have lower glucose level, ओए हंस रही है, इशिता बता. When you have lower glucose level, which hormone should be responsible? When you have lower blood glucose level, हंसते रहे. ग्लूकागॉन, हाँ, ग्लूकागॉन का काम क्या था? अभी यहीं पे लिखा था यार. What was the role of glucagon? It was responsible to add phosphate group over the enzyme or the protein structure. Yes or no? अभी लिखा था. Once a phosphate group is added, protein kinase becomes inactivated. So you had no food, you are under fasting, causes the release of glucagon that activates an enzyme called as protein kinase A. याद आ रहा है? This enzyme is responsible to add this phosphate group. Once phosphate group is added to this enzyme, this is called as covalent modification. Fine. Once phosphate group is added to this enzyme, the PK enzyme becomes inactivated. As a result, no glycolysis. अरे yes or no बोलो यार. हाँ? इसका reciprocal क्या होगा? What is going to be the reciprocal part of it? You were fasting, na? You had food. You are in well-feeded state, causing the release of insulin. Insulin is responsible for activation of an enzyme called as phosphatase. Yes or no? This phosphatase is responsible to remove the phosphate in the form of inorganic phosphate. Once phosphate group is removed, the PK becomes active. Once PK is active, glycolysis goes on. This is called as covalent modification of pyruvate kinase. I told you, na? It is very simple. It is very simple. The only thing is you have to learn is how to apply smartness in biochemistry or in this sort of regulation process. You have to apply smartness. Pyruvate kinase is a part of glycolysis, and glycolysis is needed only when either glucose level is very high and we are deficient of ATP or energy. Glycolysis should go on. Glycolysis should be stopped when we have less concentration of glucose and we are reaching ATP. यही दो condition हो सकता है. If we have less concentration of glucose, glucagon comes in, adds a phosphate group, enzyme is inactivated. Again, you had food, glucose level is high, Insulin comes in, removes the phosphate group, protein kinase becomes activated. Simple. इससे ज़्यादा कुछ नहीं है. Am I clear? Am I clear to all of you? From this topic, glycolysis, what is important for you guys is this part, regulation. Very important. Second is deficiencies. The commonest out of deficiencies from this metabolism is pyruvate kinase deficiency. Mostly seen, very rare but still common. Very rare but common amount. You know, pyruvate kinase deficiency that causes hemolytic anemia. Remember this. Why hemolytic anemia if pyruvate kinase deficiency? Anyone? Hemolytic anemia, hemolysis, there will be excessive hemolysis of RBC. Why? If there is a deficiency of PK, pyruvate kinase. Why? अरे यार तुम लोग बड़ी भोंदू हो. Yes? 120 days? Maximum lifespan is 120 days. But hemolysis, excessive hemolysis, that means the RBCs are dying before 120 days. If there is a deficiency of enzyme called as pyruvate kinase, the last enzyme of glycolysis. Why they are dying before 120 days? सही है, देखो, simple. Good, sit down. If there is a deficiency of an enzyme called as pyruvate kinase in the RBC, we know that RBC is totally dependent on glycolysis for the production of energy. Yes or no? है ना? If glycolytic enzymes are not there, like pyruvate kinase, glycolysis will stop down. There will not be any glycolysis. As a result, the production of ATP in the RBC will get hampered. Because of that, the RBC will lack of ATP. Energy रही रहेगा. अपन बचेंगे क्या? तो RBC कैसे बचेगा? वो तो माइक्रोस्कोपिक है. हमारे पास एनर्जी नहीं रहा तो हम नहीं बचेंगे. तो RBC तो माइक्रोस्कोपिक है, उसको तो मरना तय है. Because of that, excessive hemolysis. Am I clear? Am I clear to all of you? Point number 2. There is another rare but seen cases are seen. Deficiency of 1,3-bisphosphoglycerate mutase. Sorry, 2,3-BPG mutase. The enzyme that is responsible for production of 2,3-BPG in the RBC. वो वाला, वो husband wife और सौतन वाला story जो था, ठीक है? If there is a deficiency of this enzyme, what will happen? In RBC, if there is a deficiency of enzyme 2,3-bisphosphoglycerate mutase, what will happen? What will happen? Think. There will not be any production of 2,3-BPG? Yes or no? 2,3-BPG production band. As there is no 2,3-BPG, will the hemoglobin present in those RBCs are going to deliver oxygen when needed? No. Because of that, they may lead to hypoxia, leading to excessive hemolysis. Is that clear? Please. Feel free to ask or approach if any doubt you have. Okay.